And as Leah said, today I will be presenting on the research that I've been doing in Dr. Warren Chan's lab this past summer, which is on developing a morphology-based classification algorithm for identifying caveolae, clathrin vesicles, and macropinosomes on electron microscopy images. And without further ado, I'll get started. So nanoparticles is a type of nanomaterial that are very similar in scale with biological molecules. And these nanoparticles have sizes that we can control. As well, nanoparticles can deliver imaging and therapeutic agents to disease cells, such as tumors. A study by Bezadi et al. Suggests that nanoparticles can enter cells through endocytosis. Parton et al. Also supported this notion that endocytosis is a critical step in how nanomedicines can reach their intracellular targets. Essentially, these nanoparticles would enter the cell outlined in gray here on the left side after interacting with components of the plasma membrane. Now, this interaction alludes to a particular mechanism that uptakes nanoparticles into cells. In this image here, we see nanoparticles residing in an endocytic vesicle of endothelial cells. There are various mechanisms and pathways that can uptake foreign substances into cell. Each of these different uptake pathways have different vesicle types associated with them, and thus each of them also vary in characteristics. On the very left side here, we have the macropinososis pathway, which then forms a macropinosome. They're very inhomogeneous and irregular in size. And then we have the clathrin-mediated endocytosis, which then forms a clathrin vesicle. They're characterized by their thick electron-dense coat. And lastly, we have the caveolae-independent endocytosis, which then forms a caveolae, and they're very flask-shaped. Now, transmission electron microscopy allows us to do two things. First, to resolve these vesicles inside the cells with sub-nanometer resolutions. And secondly, they allow us to visualize the uptake directly, because we can then deduce the mechanism, the uptake mechanism, by observing the characteristics of these vesicular structures. In our case, we image vesicles inside endothelial cells. So as you can tell from these images, these endocytic vesicles are very difficult to characterize subjectively and distinguish from one to another, as one, for example, can easily mistake a caveolae as a clathrin vesicle. Now, this inconsistency and challenges in subjectively classifying different vesicle type on TM images is the rationale for my project this summer. So as per the previous slide, the roadblock that we're facing right now is that the morphologies of these different vesicle types is challenging to differentiate on TM images subjectively. There exists a plethora of biases and inferences which vary from one scientist to another scientist when identifying these vesicles. So the objective of my project is to develop a classification algorithm for these endocytic vesicles on TM images based on quantifiable features. So to put it simply, we want to explore if there are defining quantities that would help distinguish, for example, a macropinosome from a caveolae. And to take the project a step further, we would then like to automate this algorithm such that it would enable for consistency in assumptions and in hope to reduce data handling time when analyzing larger data sets. We care about characterizing and classifying these endocytic vesicles into different vesicle types because then it will enable us to apply the same methodology to nanoparticle-containing vesicles inside of cells. The development of this classification algorithm demonstrates an objective, impartial, and consistent methodology for identifying and characterizing endocytic vesicles in cell. To study which uptake pathways are utilized by nanoparticles, we need to first be able to characterize and identify nanoparticle-containing vesicles, so we can trace it back to its corresponding pathway. To do this, we would feed TM images into the algorithm, which will then aid us in identifying and classifying the vesicle type automatically. And thus, at the end, it can help us deduce the uptake pathway that the nanoparticle used. Vesicles and their associated pathways may have different efficiency in uptaking nanoparticles across the plasma membrane. So for example, if we find that nanoparticles exist more abundantly in caveolar structures, vesicles, we would then want to design our particles small enough to fit in a caveolae, so that they can get uptaken better with that respective pathway. And as a result, nanoparticle delivery for imaging and therapeutic agents can be increased for various diseases. We conducted a preliminary analysis to determine which important morphological features we can use to build this classification scheme. So our workflow include using an image analysis software named Fiji ImageJ. And for each vesicle, 2 different traces needed to be drawn in order for us to be able to extract our desired data. And using these traces on ImageJ, we can then obtain the vesicle's parameters. So we wanted to look at cross-sectional area, perimeter, ferrite diameter, coat thickness, circularity, and finally the contrast ratio between the vesicle coat and its lumen. What we found out was that different vesicle types do have quantifiable characteristics. And to determine the appropriate and best parameters to use when comparing between different vesicle types, we analyzed our data and conducted statistical analysis on them using the ordinary one-way ANOVA and the Tukey's multiple comparison test. So one of the parameters that we tested and analyzed is cross-sectional area. And here, if I could direct your attention to this large figure, comparing between caveolae, clathrin, and macropinosomes cross-sectional area. But more specifically, I'd like to direct your attention to the macropinosome and clathrin data. You could see that when compared to each other, they have a p-value of less than 0.0001, which means that they're statistically significant. The large differences in the data does not happen by chance, but rather they're attributable to a specific cause. This suggests that cross-sectional area is a good parameter to use when trying to distinguish between a macropinosome and clathrin. So if we have an unknown vesicle with a cross-sectional area input of over 300,000 nanometers squared, the algorithm can confidently suggest that the vesicle you have at hand is a macropinosome instead of a clathrin or a caveolae. The preliminary analysis allowed us to find out which parameters or morphological features we can rely on to help distinguish between these different vesicle types. The 4 defining parameters that we used to build the classification scheme are ratio contrast between the vesicle coat and the lumen, the ferrite diameter, the coat thickness, as well as the cross-sectional area. Using this information we have gathered in the preliminary experiment, we built this classification algorithm in this slide on MATLAB that allows for it to automatically classify a vesicle in endothelial cells to one of the 3 types that we have outlined based on their input parameters. It is very similar to a decision tree, where when used for larger data sets with many vesicles in it, it will then output the number of vesicles that has been classified under each type. So how many clathrin vesicles, how many micropinosomes, and caveolae in a given data set. In terms of next steps, to ensure the robustness of this algorithm, we are currently in the process of validating the semi-automated classification algorithm. We plan to compare the results outputted by the algorithm to manual and subjective classification of these endocytic vesicles done by users. So, to summarize my presentation, we learned today that endocytic vesicles on electron microscopy can be characterized and classified quantitatively, which provides a more objective methodology compared to the one used in the field right now, which has been done subjectively. So with that, I conclude my presentation and I will answer any questions that you may have. Thank you.
Thank you, Wanda. Wanda, I was curious, the caveolae and the clathrin data seems to overlap a lot. Can you speak to how these can be differentiated and how you can know whether one vesicle is truly one or the other?
Yeah, for sure. Thanks for the question, Leah. So the way that we see this algorithm, it's kind of like a sift. You go step by step. It's a step-by-step process. So in the way that it works in the classification algorithm, in the flow, is that we will go through the contrast ratio first. So then differentiating caveolae and clathrin based on that parameter. And then we go to the next step and differentiating between them, for example, through coat thickness. As it is an algorithm, it is not yet perfected. And that's what we're doing with the validation step to see whether or not we can improve with the algorithm that we have right now. So right now we're just testing it in terms of how we can make it more robust and accurate enough so that we can use it for applications.
Great, thank you. And then you mentioned inter-rater reliability with the algorithm. Could you speak more about that?
Yeah, for sure. So inter-rater reliability refers to the consistency of decisions or classifications between two individuals, and it contributes to the validity of the data. So when two people annotating, for example, with a differing degree in expertise or experience, when they're, for example, looking at a TM image, it may result to different outcomes. However, the algorithm allows us to remove this knowledge imbalance entirely so that all decisions is all dependent on the network, which the algorithm is based upon.
Great, thank you very much.